Hello everybody, my name is Shivan and in this video I am going to explain briefly about Hybridoma technology. Before going further, let's take a quick history lesson. This technology was originally developed by two biologists named George and Caesar in the year 1975, and in 1984 they've also received a Nobel Prize for their research. What they have done is that they have successfully managed to create a hybrid cell which has the ability to fight against antigens. Now let's go through the principle behind this technology. This hybrid cell has the ability to produce antibodies, which was originally believed to only exist in B cells or spleen cells or plasma cells, and at the same time they can also replicate themselves continuously without dying, like myeloma cells. So by combining these two desired qualities of these both cells, this technology ensures large antibody production. This is called hybridoma technology. Why we need this? As I said before, that the antibodies are only produced by the spleen cells, but the problem is that they have very short lifespan. We are talking about few weeks. This takes very long time to produce enough antibodies to develop the cure to certain deadly diseases. Now, what is a monoclonal antibody? A monoclonal antibody is an antibody made by cloning a unique white blood cell. All subsequent antibodies derived this way can trace back to a unique parent cell. Monoclonal antibodies has the ability to bind themselves to a specific epitope. These are specially used for treatment of cancer cells and can also be used as antivenom. So this is how a hybridoma cell is formed, by fusing a plasma cell and a myeloma cell. This hybridoma cell can produce antibodies and they are immortal. In order to successfully produce a hybridoma cell, we need to follow this 5-step procedure. The first step in this procedure is the immunization of a specific mammal. This is done by injecting antigens into the mammal intravenously into their bloodstream. Then their spleen activates B cells, which then produces plasma cells. Then these plasma cells needed to be isolated from spleen. So this is how immunization takes place. Let's assume that antigens are directly injected into its bloodstream. Then its B cells produces plasma cells, which has the ability to produce antibodies. The next step is the isolation of myeloma cells. Myeloma cells are abnormal plasma cells that build up in bone marrow and form tumors in bones. So normally these are cancer cells and they need to be isolated from bone marrow. As I said earlier, that the fusion need to take place between plasma and myeloma, and this can be done by two methods. One is in the presence of medium and another is by electrofusion, but fusing through the polyethylene glycol solution is popular. This is how fusion of plasma and myeloma takes place in the presence of polyethylene glycol, which results in the production of 5 different hybrid cells, as you can see here: fused plasma, fused myeloma, hybridoma, unfused plasma, and unfused myeloma are the 5 cells that are produced by the fusion. Before multiplication and production of antibodies, we need to create many more hybridoma cells, and for this we need to synthesize new copies of DNA, and for that it requires synthesis of nucleotides, and this can be achieved by two different pathways. One is salvage pathway and de novo pathway. In salvage pathway, it requires degraded path of old nucleotide to produce new nucleotide. In de novo synthesis, it synthesizes an entirely new nucleotide with the help of molecules of certain sugars and amino acids. As you can see that this is how DNA replication takes place from mother cell to daughter cell in hat medium. But in hat medium, cells cannot be synthesized by de novo synthesis due to the presence of aminopterin in HAT medium, which blocks dihydrofolate enzyme, which is necessary for their synthesis. So our only option is to perform synthesis using salvage pathway, but it must require the presence of HGPRT enzyme, where hypoxanthine and thymidine are used as precursors. So these are the 5 cells that are produced by fusing spleen and myeloma cells. As you can see, only few cells has HGPRT enzyme. The rest don't have it. Fused myeloma and unfused myeloma don't have HGPRT enzyme, so they cannot survive in HAT medium. Fused plasma and unfused plasma have HGPRT enzyme, but they have very short life, which leaves us with hybridoma cells that have HGPRT enzyme from spleen, as well as they also have very long lifespan too. So hybridoma cell is the only cell that can survive in HAT medium. Thank you.